There is a mathematical model, originally designed to describe chemical reactions in a tank reactor, that, when you tweak two numbers, produces the stripes of a zebra, the spots of a cheetah, or the maze on a pufferfish's skin. That model is called the Gray-Scott reaction-diffusion system. It's a type of Turing Pattern, named after Alan Turing, who in 1952 proposed that chemistry alone, governed by the right mathematics, could self-organize into complex biological form, independent of genetic instruction. In this video, I'm going to simulate the Gray-Scott model using Python across 3 different parameter sets and ask, can a set of differential equations genuinely replicate what we see in nature? And if it can, what does that actually mean for our understanding of the natural world? So here's the Python code. See here, I got the equations from a scientific paper, and these are the main parameters here. So I'm going to be changing f and k, and I'll explain how it works later. So if I run it with these settings, with f as 0.027 and k as 0.06, this will give me the spots pattern. So as you can see here, kind of started off as C shapes, and then it splits up. These sort of lines, and they split into individual dots. And this is similar to what we get with a cheetah's coat. So you even get like some slight differences in size between the spots, which you definitely get on the cheetah's coat, although there's is more pronounced on the cheetah. But it's pretty similar likeness. And now with f set as 0.03 and k remaining as 0.06, if I run this, I should get a mixture of spots and stripes. Again, it starts off in a similar way, but you can see now I'm getting a lot of more lines that are remaining instead of splitting into individual spots. And this is just like what you see with a whale shark. So you get a lot of spots and stripes that are more sparse towards the back of the animal, and then as you go towards the head, it gets more clustered and concentrated. You get more spots at the head of the animal, so the pattern kind of changes as it goes along a little, but also very similar. You get some spots in between the stripes. We get a little bit of that here. If we had more iterations, we'd probably end up with something even more similar. And finally, and most importantly, if I have the settings like this with f as 0.029 and k as 0.057, I should get the labyrinth patterns. So let's run this. And wow, and you can see straight away it starts forming these maze-like patterns. And what does this look most similar to in the animal kingdom? Well, initially I looked at the giant pufferfish. This is what I based my original study on. And you can see it's pretty similar to the skin patterns on the pufferfish here. Although there are some marked differences where the skin of the pufferfish seems to have sort of own individual sections. The pattern is a little bit different. But there's still a good likeness. If I also compare it to brain coral that you get in a lot of tropical regions of the world, you can see it has a very good resemblance of the pattern. Also, I later discovered that there is another fish that has an even better likeness with the simulation labyrinth pattern, and it's this. This is called the maze rabbit fish. Literally great name, because it does very much have the maze pattern. And you can see, I think this is probably the best resemblance that I've seen in nature. Most reaction-diffusion models, like the classical Turing patterns, use two explicit morphogens: an activator that promotes its own production at short range, and a fast-diffusing inhibitor that suppresses the activator at a longer range. The Gray-Scott model is different. There is no explicit inhibitor. Inhibition happens passively, by the activator depleting its own substrate. Think of it as grass and rabbits in a grassland. The substrate is the grass, and it grows continuously. The activator are rabbits that eat the grass, and they reproduce, and they're able to diffuse or spread out faster. And then you have the two parameters that I changed in the video, which were F, the feed rate, which is the rate at which the substrate, the grass, regrows, and K, the kill rate, which is the natural die-off rate of the activator, which is the rabbits. The pattern is what's left behind. Where rabbits have stripped the grass, you get bare patches, and it's the geometry of those depleted zones, shaped by the relative speed of diffusion and the FK ratio, that determines whether you get spots, stripes, or a labyrinth. Meaning, you don't need a blueprint encoded in the genome for every stripe. You just need the right kinetics. The pattern is an emergent property of the reaction. When compared side by side, the reaction-diffusion labyrinth simulation is a compelling match with the skin of the giant pufferfish. Interconnected maze-like bands, consistent pathway widths, and continuous connectivity. The structure is right. But look more closely at the pufferfish. The edges are sharper, the bands, particularly the black areas where the substrate is depleted, vary slightly in width. There's what looks like closed off sections, more randomness, and less periodicity in the real animal. The simulation is too clean. That's not a failure of the model. It's a signal that additional biology is operating on top of the mathematics. In fish, the cells doing this work are called chromatophores. They are pigment-containing cells, named for what color they carry. Melanophores for black, xanthophores for yellow. These cells don't simply diffuse across the skin like molecules do. They migrate actively, and crucially, they talk to each other through direct physical contact via long cellular protrusions. And here's where it gets interesting. When you look at how these protrusions actually behave, you start to see the reaction-diffusion logic re-emerge, but instantiated in cell biology rather than chemistry. At short range, melanophores and xanthophores mutually repel each other, and if forced into close proximity, they can destroy each other. The xanthophores mediate this through short protrusions. From the pattern's perspective, this local repulsion is actually a positive feedback loop. It keeps melanophores clustered in their own stripe, reinforcing the black band. At long range, the relationship inverts. Melanophores extend long protrusions deep into xanthophore territory, and depend on the presence of those distant xanthophores for their own survival. Remove the xanthophores, and the melanophores die. This long-range dependence acts as a negative feedback loop on the pattern. It puts a ceiling on how far a black stripe can expand before its leading edge loses contact with the xanthophores it needs. This is the short-range activation, long-range inhibition logic of a Turing system. But the diffusion coefficient in the equation maps onto protrusion length in the biology. A longer protrusion reaches further, functioning as a proxy for a molecule with a high diffusion rate. The mathematics is the same. The physical substrate is completely different. And that's the deeper implication of the simulation being too clean. The Gray-Scott model captures the logic of the pattern, but in living systems, that logic is implemented through cell biology, developmental signaling, and evolutionary history, all operating simultaneously. The mathematics is the skeleton. The biology is everything built on top of it. There's a compelling hypothesis about where the labyrinth came from, evolutionarily. Labyrinthine patterns on fish show a strong association with hybridization events between ancestral species. The proposal is that blending two simpler spot patterns, dark spots on a light background, from one ancestral lineage, and light spots on a dark background, from another, can generate a labyrinthine structure. This has been demonstrated in silico and partially verified using salmonid hybrids. It's still a hypothesis, but it's a testable one. Selective breeding of different pufferfish species that express the simpler ancestral phenotypes, dark spots on a light background and light spots on a dark background, then crossing them, could directly probe whether the combination produces the labyrinth in the offspring. What's striking about this hypothesis is that it makes the labyrinthine pattern not just a mathematical artifact, but an evolutionary signature, evidence of a hybridization event written into the skin. Two numbers, f and k, tweak them within a narrow range, and the same set of equations produces the coat of a cheetah, the flank of a whale shark, the skin of a pufferfish, or the surface of brain coral. That's not a coincidence. That's convergence on the same mathematics across entirely different biological lineages. And it's worth asking, what are these patterns actually for? In predators and prey, the answer is often camouflage or signaling. Natural selection has clearly acted on these patterns. But the same mathematics also produces the labyrinthine surface of brain coral, and coral isn't hiding from anything. The reaction-diffusion system didn't necessarily have an original purpose. It just produces stable periodic structures wherever the conditions are right. Evolution happened to find those structures useful in some lineages, and kept them. But the mathematics came first. What the Gray-Scott model reveals isn't just that nature is orderly. It's that the order is deeper than genetics. The genome doesn't micromanage every spot and stripe. It sets the parameters. The math does the rest. And that raises a question that I think is genuinely profound. How much of biological form, from pigmentation to digit spacing to the folding of the brain, is not encoded but solved? Solved by the same kind of self-organizing reaction-diffusion dynamics, converging on the same patterns again and again, because those patterns are what the mathematics produces when the conditions are right. Nature doesn't just follow the laws of physics. It computes with them. If you've watched all the way to the end here, firstly, thank you, And secondly, please like and subscribe to help me out. Because, well, I'm a broke science student trying to start a YouTube channel. And trying to gain traction on YouTube can be really tough, especially for new channels. So I'd really appreciate it. By the way, I've included the Python code in the description for anyone that's interested in playing with these simulations. And also, check out my other videos on my channel. Or just come back for the next one. See ya.